Thank Paul. It is my honor to be here to talk about some of our work in clonal evolution in cancer. I'm going to introduce a tool that we developed for inference, visualization, and interpretation of clonal evolution, and I'm going to demonstrate some application in leukemia and colorectal cancer. Studying tumor heterogeneity and clonal evolutions have provided us with many useful insight into the mechanism of disease. To exemplify this, I'm showing here a recently studied AML patients with whole genome sequencing and targeted sequencing. We discover a very complex clonal structure from this patient from the primary and relapse samples. To visualize the clonal evolution and track the cellular fraction change of the clones over time, we create this fish plot to fish plots. Each of the shape here representing a clone, and how much of the space occupied represent the cellular fraction or prevalence of the clones. And the shape is nested inside the other one, indicate the parental and child relationship. What we can see or learn from here is that the very low frequency subclone in the primary then escape chemotherapy, continue to evolve under chemo treatment, and subsequently gives rise to additional subclone. Together, they define the relapse. In order to reconstruct this clonal evolutions, a two-step approach is often employed. First, we want to identify the clones by clustering the variant based on the prevalence. So the variant with similar prevalence are being clustered together. The prevalence can be measured by the variant allele frequency, VAF, or by the cancer cell fraction, which is the CCF, can be calculated twice of the VAF if there's no copy number alteration. As you can see here from this AML patient, cyclone was used to cluster, and it's identified 6 different clusters. Each of them we call the clonal marker for the corresponding 6 clones, and including a founding clone, another share clone, and a few other clones are private to only 1 sample. Now the next step is to order or nest the clones to identify the parental-child relationship or the clonal evolution model based on the prevalence estimated based on the cluster. This task is called clonal ordering, and technically it just enumerates all possible clonal evolution models that satisfy some biological constraints. There are 2 constraints that governing the clonal ordering step. The first one being the sum rule. It can be described by several equations. The first one saying that the CCF of the cluster has to be equal to the CCF of all the clones that carry the mutation in this cluster. This is biology. If you apply this recursively down the clonal evolution tree, you can kind of convert this into a more computable equations where you have the CCF of the clone, the descending clone of the clone that you care about, and reflect by the CCF of the cluster of the direct subclones of the clone that you're calculating. Now this allows you to calculate the CCF of the clone based on the CCF of its cluster, and then the CCF of the cluster corresponding to direct subclones of that clone. Since CCF has to be non-negative, then the prevalence of the parent clusters must be greater than or equal to the total prevalence of the child clusters. And the second rule is cross-rule. The parental and child relationship of the clones must be maintained in all related samples from, for example, from the same patients. Now the problem is this clonal ordering step is often done manually when you look at the point estimate of the prevalence of the clones. And the issue here is the sum is very oftentimes being violated by the point estimate, because the point estimate of the CCF or VAF is oftentimes very uncertain, This could be due to the inadequate depth, random errors, or analysis error. Oftentimes, when you have single biopsy, the chances you're getting only, or the chances you're not seeing a parental clone is pretty high because of intratumoral heterogeneity. And that creates the CCF estimated clone. With 50% of the time, you get a negative estimate, and that is the 50% chance of having a sum rule violation in one samples. Now, back to this AML case again, you have a subclone number 3 here. This cluster actually has a slightly higher VAF estimate or CCF estimate compared to the founding clone. Fortunately, these two clusters are quite distinct in the primary, so that provides you better resolution in the samples that will govern the clonal ordering. However, this is a simple case with 2 samples. If you have more samples, it's getting more complicated, and there's a need for an automatic method that can control for or deal with the violation of sum rule simultaneously in multiple samples. That's why we developed Clonal. Using this equation with sum rule, Clonal can estimate the confidence interval of the clone CCF using a bootstrap resampling techniques. Now, this will allow for the test of sum rule violation, satisfaction or violation. Also allows for determination that if clone exists in a sample or not, and also allows for the classification of the clone into a founding clone of the sample or the subclone, and that further enables the interpretation of a clonal seeding. Another challenge is how to very quickly generate very intuitive visualization for the clonal evolution, because clonality analysis is a very iterative analysis with multiple steps, and you oftentimes have to introduce model and refine it and over again. So we also, Clonal is able to automatically generate a few plots that have been very helpful. We replaced the scatter plot cluster by the zeta plots that now allows us to look at and compare between samples and between clones. And it's allowed overlaying more samples into this without changing any, without making it any more difficult. But more importantly, Clonal was able to generate bell plots for individual samples that mimicking the fish plot, and also automatically generate the tree that is annotated and helping with a better interpretation of clonal seeding. So now I'm going to explain how this is going to be is helpful in the AML case. So this is the model inferred by Clonal for the AML case. One thing is if a clone is not found in the sample, is white out in this bell plot, you can immediately recognize this from this relapse samples. The clone number 3 actually being the founding clone of this sample, because it's the first clone you see that says non-zero cellular fraction. And then you can make a connection that this clone coming from the primary in the clonal seeding model. And this is identically reflected in the tree. In the tree, you have the clone as a node annotated with the sample if the clone is found to be non-zero cellular fraction in the sample. A star indicated that the clone is a founder clone of the samples. Now you can look at here and see that the clone number 3 is the only one that's shared between the primary and relapse. And it is found in relapse, a subclone in primary. Again, this is a monoclonal seeding from the primary to the relapse. Very intuitive. So to this point, I have shown in how Clonal has been helpful for improve the analysis and interpretation of a very well-characterized AML case. I'd like to share with you now some of the preliminary results that in our efforts to understand clonal evolution in colorectal cancer. So in this project is the discovery phase. We start with whole genome and whole exome sequencing of multiple samples, including a primary sample, several metastatic sample, and xenograft sample from one individual. We have actually 10 patients. And in the validation phase, we use targeted sequencing to in the discovery sample, and also include additional multi-region sample from the primary. And we cluster ran using Cyclone, using Clonal to for the clonal evolution model. This is a representative case. This case is a synchronous sigmoid colon cancer. We provide 14 different regions from the primary. And originally have 2 distinct metastases from under the liver. And one of them went through the xenograft in the mouse, and we have an additional xenograft sample after 8 weeks of engraftment. This is the clustering result using Cyclone, identify 14 different reliable clones or cluster. It's important to notice that some rule is violated very frequently. Almost all samples have some rule violation. If you look at only the first 3 cluster, already 5 samples violate the sum rule. Believe me, this is real. If you feed this directly to Clonal, this is what we got. Clonal evolution for all samples individually. They are matching across all samples. And you can notice that they are all different in the structure of the clones. How many clones are there in those samples are different. It's important to notice that among 14 clones, there's only a few are detected in only 1 sample. If you're lucky, you get 5. If you're not, typically you get 2 or even 1 clone in 1 sample. This is because of a great intratumor heterogeneity that was found in the primary sample and by exhaustive profiling of this primary tumor, we're able to see this. Now it is possible to now looking at this and infer what clones drive or seed those metastases. But let's do it in another more simple way by looking at the clonal evolution tree. It's immediately recognizable that this founding clone from the tree branch up into very distinct branches. The first branch is a metastasis branch that involves all the metastasis sample. The non-metastasis branch consists of only clones that are found in the primary tumor, very clear. So these 2 clones are different. There's something going on here that makes them different. One was able to metastasis, one not. And then further, if you want to look at the metastasis seeding, there's a clone right here, number 8, that's which found to be a subclonal level in primary section 13. And then being found for the MET number 2 and also another primary section or region that is a monoclonal seeding model here, clone 8 moved from section in primary to the METs. And this is important. Notice it's captured by only 2 primary regions. So intratumor heterogeneity plays a big role here. This MET1 has a founding clone number 11. It was not detected elsewhere. So this is challenging. It might be that we missed these founder clones in the primary such that we don't see it. But we have 14 samples, so we think, well, we probably exhausted the primary tumor. So the other explanation, probably the intratumor heterogeneity in this MET as well, that prevented us from seeing the actual clone that found this. And that might be clone number 2 here that was detected in the primary region number 7 at subclonal level. So that might be another single cell or monoclonal seeding as well. So it's very intuitive by looking at the tree here and figure out the clonal seeding model from primaries to metastasis. Now, if you want to focus only primary and metastasis relationship, ignoring multi-region and intratumor heterogeneity in the primary, you can even, or before that, actually there's one other subclone survive in the xenograft and evolve. So if you don't care about intratumor heterogeneity in the primary tumor and focusing on primary and metastasis relationship, you can further merge those multi-region from primary into form a big tumor here that represent all the trees that was discovered from the primary regions. And then if you overlay this on a clinical timeline, all the sample overlay, and then make the connection between the primary and the metastasis, then you kind of now seeing the very complex case now being very simplified. The interpatient is very simple, and then it's bringing more, we can bring more in-depth interpretation with chemotherapy involved. Now, we haven't done this yet, but it's very easy, or not easy, but it seems very intuitive to bring in some of more of the clinical data. So single cell seeding, 2 mets, and then you have a xenograph, actually a subselection of a clone number 9 in a xenograph, giving evolving gives another subclone. So with that, I'd like to summarize. Accurate construction of tumor evolution requires exhaustive profiling of intra-tumor in intertumor sites. In this context, Clonal is a useful tool for error-tolerant inference, visualization, and interpretation of clonal evolution in many samples. Clonal enabled the discovery of relapsed clones in acute myeloid leukemia. It simplified the interpretation of metastatic colorectal cancer in very complicated case. And with that, I would like to thank all the people behind this project, the funding agency. The Chris Maher Lab is where I'm training as postdoc. Stephen Fo is the graduate student who wrote some code for the parametric bootstrapping model. Brian feels happy with Julie, Matthew, Elizabeth has been working really hard to get good data to me. And Brian Y was the person who did an initial analysis for the CSC projects when I took over. Chris and Malachi has been a great collaborator so far. Clonal is available now at GitHub. Thank you very much. And
thank you very much. Great talk. We have time for some questions.